I'm Hugh Rosen, and I'm a professor in the Department of Molecular Medicine, and I'd like to talk about COVID-19 and the riddle of the Sphinx, how we've discovered some risk factors that inform intervention in COVID-19 with Osanamod, a drug discovered and first synthesized at Scripps Research. What is Osanamod? Osanamod is a selective and well-tolerated oral treatment for serious autoimmune diseases that was invented and first synthesized at Scripps Research. It's an effective single daily dose treatment for relapsing, remitting multiple sclerosis and is marketed worldwide as the drug Zeposia by Bristol Myers Squibb. It is currently under FDA priority review for ulcerative colitis. The first description of the mechanism, namely sphingosine 1-phosphate receptor agonism, was in a science paper from my lab in 2002, and this was the first new chemical entity that began life as a hit from the NIH library and the MLPCN screening center at Scripps, Florida. It's a disease-modifying treatment which shows multi-step interdiction of the disease process that inhibits both acute attacks in multiple sclerosis and in ulcerative colitis, and also prevents long-term tissue damage. Why this is a disease-modifying therapy is because it interacts with the disease at multiple levels. In the immune system, it alters the movement of lymphocytes and their migration into the tissues where they cause damage. At the level of the blood, it tightens endothelial barriers, improving vascular integrity. In viral and inflammatory diseases, it suppresses cytokine storm, both in pandemic influenza and, as I shall show you, in infections with other viruses, including SARS-2 COVID-19. It suppresses key regulatory cytokines, such as interferon alpha. And so therefore, this mechanism is useful in other autoimmune diseases, as well as viral diseases that are characterized by aberrant cytokine signatures. COVID-19 infection, of course, is well known to all of us and has two distinct phases. It has the infectious phase, where virus replicates and directly damages cells. And in most people, this phase resolves without proceeding to the high-risk phase of the disease, which is, of course, the inflammatory phase. The inflammatory phase is usually delayed 7 to 10 days after the initiation of infection and is characterized by immune-mediated damage to tissues, most especially the lung. There is strong evidence in COVID patients for a cytokine storm, or what is called the cytokine response syndrome, leading to a leaky lung. There are associations with common pre-existing conditions, such as obesity, diabetes, and metabolic syndrome, that seem to elevate the risk for the inflammatory phase. And we do not yet have a full understanding of the mechanisms involved. So in a fascinating study from Mafia et al., looking at around 140 consecutive Italian patients hospitalized during the peak of that initial COVID outbreak in Italy in May and June of 2020, they discovered that low high density lipoprotein was a risk factor for poor prognosis. So patients that had low high density lipoprotein in their blood had a poor prognosis and showed increased admissions to intensive care units and had a higher mortality. Normal patients, or patients with HDL in the normal or high range, were not admitted to hospital, were not admitted to ICU, and had a distinct survival advantage in response to COVID infection. So why does this happen? And what does it inform us about potential treatments for cytokine response syndromes in the inflammatory phase of viral infections? One of the key features is that high-density lipoprotein is the reservoir for sphingosine 1-phosphate. And as I explained with Osanamod, sphingosine 1-phosphate acts on its receptor, shown here in blue, to maintain the endothelial barrier and the integrity of blood vessels, inhibiting leak into the lung. In addition, it acts on the plasmacytoid dendritic cell, that is the key orchestrator of cytokine storm. And we have direct evidence that shows that the S1P receptor is responsible for down-modulating cytokine storm and thus protecting. So there is the potential for high-density lipoprotein to protect in multiple ways, but also inform how we can use drugs to enhance the effects of HDL and therefore put patients into lower risk populations. So 98.5% of blood sphingosine 1-phosphate is bound in HDL. So why then is low S1P an independent risk factor for poor COVID-19 outcome clinically? So this is what we know about sphingosine 1-phosphate. Agonism, activation of the sphingosine 1-phosphate 1 receptor suppresses cytokine storm. So here we see in the absence of activation of the sphingosine 1-phosphate receptor, we see around 4,000 picogram per mil of interferon alpha being produced. And this is the black bar. In the presence of the S1P receptor agonist, closely related to Osanamod, called CYM, shown in red, you can see the complete suppression of interferon alpha production by activation of the sphingosine 1-phosphate receptor. And this can be achieved both with pharmacological agonists as well as the natural ligand sphingosine 1-phosphate itself. How does it do this? Sphingosine 1-phosphate binds to its receptor, shown here in red on the plasma membrane. It drives the receptor into an intracellular compartment called the endosome, where signal termination occurs. The receptor is then diverted to another organelle called the lysosome, where it is degraded into its component amino acid parts, thus terminating the response to sphingosine 1-phosphate. At the same time as the sphingosine 1-phosphate receptor moving into the degradative pathway, it drags the type 1 interferon receptor into the same degradative pathway, and en passant terminates the signaling through the interferon alpha pathway and diminishes the amplification of interferon alpha, which requires an interferon alpha receptor auto-amplification loop. So does this work in SARS-CoV-2 infection? So here, John Taharo infected hamsters with the SARS COVID-19 virus, and we showed that both body weight change and the survival of hamsters was enhanced by treatment with Osanamod, shown in red, compared to the vehicle, shown in black. Furthermore, when we looked through RNA sequencing, we found that T cell marker genes, the upregulation of T cell marker genes, as well as B cell marker genes, were suppressed in the Osanamod treated infected hamsters compared to vehicle. In addition, the interferon alpha response, the cytokine storm, and inflammatory responses were suppressed by Osanamod and and upregulated under vehicle conditions, showing that setting the sphingosine 1-phosphate signaling rheostat protects from the cytokine response syndrome in SARS-CoV-2 infection. We know that low levels of high-density lipoprotein in blood are associated with obesity, diabetes, metabolic syndrome, and lack of exercise. It is an independent severity predictor for the inflammatory phase of COVID. It predicts ICU admission and enhanced mortality. The mechanism of low high-density lipoprotein risk appears to reflect the role of HDL as a reservoir for sphingosine 1-phosphate, which in turn is required to enhance endothelial integrity and limit cytokine storm. And sphingosine 1-phosphate signaling tone can be successfully augmented with Osanamod. So Osanamod suppresses the cytokine response syndrome. The suppression of cytokine amplification is a shared mechanism in immune-mediated tissue damage. We know in humans that it is effective in the treatment of multiple sclerosis, ulcerative colitis, and is currently completing phase 3 for moderate to severe Crohn's disease in both children and adults. It is effective in animal models looking at immune-mediated tissue damage associated with viral diseases, such as the cytokine storm associated with the pandemic influenza H1N1 of 2009, respiratory syncytial virus or mouse pulmonary virus. It is effective against SARS-1 and also against SARS-2 COVID-19. And there is currently a clinical trial looking at the role of Osanamod in the effects on admission to ICU units from COVID infection in Canada. So we're optimistic that modulating this feature of sphingosine 1-phosphate signaling can be protective, but we also think en passant that it explains why patients with obesity, diabetes, metabolic syndrome, and low high-density lipoprotein are at increased risk for ICU admission and mortality from COVID-19 infection. And I'd like to thank my collaborators, Ed Roberts, Miguel Guerrero, Steve Brown, our co-inventors of Osanamod, Nora Leaf and Lucine for the cell-based experiments, and John Taharo, Jerry Brzak, and Namir Shabani for the hamster experiments with COVID-19.